The theme of the course is I want to try and help you, and I want you to help me to try and formulate what the overarching, most important working assumptions of biology are now and what they were. Because I think if we do this homework just a little bit together, you will find that the working assumptions of biology have not changed, even though they claim that we have made progress. We actually haven't. We're actually stuck in ideas that have been sort of assumed to be confirmed. And again, I'm kind of just wheeling here off the cuff. First of all, I want you to make sure that you understand what a working assumption is. I'm assuming most of you are not from Pittsburgh. Remind me one more time, Rick? Rick is from Pennsylvania, so he'd probably be the only person who laughs about this stuff. Or are you also from? No, okay. But if you come to Pittsburgh and you rent a car and you go on the street and you have some working assumptions about how people are going to behave, you're going to be disappointed. The first one is that because it's Pennsylvania and because we have a lot of hills, we don't like to worry about wasting our brakes to come to a complete stop. And so you might come from a place where the communication is that the first person who actually comes to a complete stop and their wheels stop turning is the first person in line. And so if 4 people come to a stop sign at once and my wheels stop first, I know I can go because you're still slowing down. That's not how it works in Pittsburgh at all. Another thing that you might not realize in Pittsburgh is that if somebody moves to the right, that doesn't mean they're going right. It usually means they're going to go left. Most people in Pittsburgh drive as though they have a boat or a trailer behind them. And so when they pull up to a left-hand turn, they come around to the right and come around. It's also because a lot of turns have pitch corners, and so you got to make sure you don't hit the hole that is actually right where the corner ends. Another really interesting assumption that you can have about Pittsburgh is that when somebody's at a left turn light, they're not going to go in front of you because they don't have the right of way. But actually in Pittsburgh, the first person does. And so if you pull up to a light where there's a left turn lane and the lights go green, you should expect that person without any real urgency to just go in front of you. And if you're a person who likes to hit the gas like that, it'll be a very shocking, like, what are you doing? And they'll look at you like, well, but that's a Pittsburgh left. And so these working assumptions, because these people went to high school here, because their parents went to high school here, because they all call each other Jagoff, they drive around with these assumptions, and they're very good working assumptions. When you see somebody go right, you know they're going left. When you see somebody there, you know they're going to go, so you wave to them, and now you get extra credit. But in biology, I think the working assumptions are something that because students have to go so hard, run so fast to get to what they are told is the edge of understanding, they just take on a bunch of working assumptions of their mentors, a lot of working assumptions of their high school teachers. I apologize for not looking over here. And those working assumptions, I don't want to ever imply in this course that I understand the extent to which those assumptions are put in place on purpose, or whether it's more of a lemmings kind of thing where the more people get the bad idea, the more the bad idea spreads, and now here we are. But I would like to start with the context that there are people in the world who are governing us under the principle that that happens. That if they convince a bunch of people that this is a good idea, then they know what people do. And you can convince people of bad ideas, and they will go along and do that too. Obviously, we get told this story when we're a kid of the Pied Piper. And so, while we are here as adults, I think I want to start with this idea that we should not take for granted the working assumptions that we have been given. And if you try to keep it as non-political as possible, that working assumption might be, for example, Donald Trump is in control, and because we voted for him, then Donald Trump says things and people do it, and that's why we're where we are. But that doesn't really make sense if what Donald Trump is doing is crashing the boat, and there are all these people who have been helping to drive the boat for 20 years, but I don't know what to do because the guy says we have to crash the boat. It doesn't really make sense, but I don't want to go there. I want to do this with the biology, because the biology is really where I think the long generational plan is. And so the other story that I'd like to come to and then throw back at as I try to get this started is the idea of my dog being very smart and also not very smart. Thank you guys very much for coming. I'm sorry I'm starting late, so you're actually just barely not even missing anything. It's kind of convenient. The Zoom meeting is here. I just told the Zoom meeting that we're late because I'm the only guy doing this, and my wife helps me and my son helps me, but all of the things that you would see, give you an example that you guys might know. Charlie Kirk does not show up and set up his own cameras and set up his tent and everything happens. Vans of shit came with him. People that make full-time salaries are behind him. And so you can't see him as a phenomenon like you're seeing here. I am a guy who at best can make a shitty omelet. That's it. I mean, I'm a real dude, and this is what I can do too. And so it's very different. I'm so freaking happy you're here. I'm going to have a lot of time to talk to you, but I just want to get the thing rolling. I believe, number one, it's important to start thinking about how we are governed, and I've said this already before. We are governed by people. I've made this point again with you. We are governed by people that are not internet phenomenons. That the outdoor boys, if you've ever seen this is a guy who lives in Alaska, and he's got 2 sons, and he took them out into the wilderness and made these GoPro videos, and he became super famous. There's like 4 of those people. Everybody else is a staff that has said, put their arm around somebody and said, you know what, you're pretty attractive. You can actually speak. You read really well. I think I could do something with you. Do you have any ideas of what you'd like to do? And then somebody says, well, I've always wanted to be a newscaster. Great, I got it. Are you liberal, or are you, how do you feel about that? And this is how we are governed. The question is how far are you willing to consider this is a possibility? Are you willing to consider, for example, that somebody like Tucker Carlson took that job a long, long time ago, that even Donald Trump or Roseanne Barr took that job a long time ago, not even knowing that the script that they were given was maybe not the most patriotic script. But somewhere along the line, they're getting a lot of money with a lot of people behind them. And remember, when I go home, I got a son who says that I'm Joe Bart, but serious. He laughed. So that's what I've got at home. I don't have a staff of people telling me that you're a genius. I've got Christy telling me that. I have Alexia telling me that, and that's about it. That's all I got. If I didn't have those people, I would be working somewhere at Home Depot right now, and this would be lost. And I'm not saying that this is a miracle. What I'm saying is that if you can take it with you, you don't have to give me credit. Even better would be for you guys to be able to go up to your doctor or your biology teacher or whatever it is next year when you go to university and ask the questions that make people go, oh shit, I didn't think about that. And these are the questions. That's what we're going to try to get to. We're going to try to get to the working assumptions that really, when you get to them, you can point this out. So, getting back to where I was before our last friends came in, I want to also give you a working metaphor by which you can imagine how we are governed. And so I'm taking a big risk here because you could think this is really dumb. But my dog Mike has taught me a lot about myself and dogs because I've never had a male dog, and this is my first male dog, and I got him because I thought he was going to be really great. But it turns out that he's a total guardian dog who would really like to kill people. And he considers white vans and blue vans to be terrorists. So it's taken me a long time to realize how much of a mistake we made by trying to get another dog to keep our old dog happy. But Mike has taught me a few things. The first thing is that Mike is working. And if you want Mike not to work, you have to tell him not to work. And so, for example, I used to just let him lead me to a person. And if that person was a male, then he would just bite him. And now I put him behind me and I shake that guy's hand, and now he doesn't do it because I'm not asking him to screen this person for me. Now, I'm just giving you a few examples of dog behavior. One of the best ones that Mike exhibits is that if you get him off the leash or you open the door, he's gone until you find him somewhere else and open the door of the car, and then he'll jump in. So he's a terrible dog to let out. But if he has his leash on, you put the leash on the ground and you click it, he thinks he's caught, and so he won't run away. You can leave him out on the porch with the leash not attached. And so he makes assumptions too. He actually assumes that if there's a line on his neck, he can't run into the street, so he won't do it, even though he can. But if you take the leash off, he'll run. Now, I want you to imagine the problem Mike would have after walking the park with me for many, many years. He's 2 years old this weekend. What kind of problem would Mike have? And I want you to start answering. Just give me some things that pop to mind. If Mike was the equivalent of a eugenicist, and Mike was smart enough to say to himself, I wonder if I could convince dogs, smart dogs like me, to make puppies. And don't at all worry. And if I did that, so in other words, do you see what I'm trying to say here? If a dog was going to try and breed their way out of servitude, Mike's got this idea that if I could just find the smartest female, we could make puppies, and because he's almost free. He looks at me all the time. He argues with me all the time. He's like, here, I don't know, I'm just staying around because you're cool. But otherwise I would be gone. And I really mean this. Like, this is a dog that's always kind of, dealing with, I'm here, but I don't really want to be. And this is a real thing. So if he, had this idea in his head, what would the task be? Can you imagine how hard that task would be? Because remember, every time Mike goes out in the street, who was that? Oh, wow, that's a female. Oh, cool. Wow, she was just here, like last week. Oh, yeah, wait. But he's still got this plan in the back of his head. He's got this plan in the back of his head that he wants to change dogs by finding smart ones and breeding smart ones. And so after a couple years, he's figured out that golden retrievers are pretty smart. But the small ones, the small dogs, oftentimes they're just worthless. So we're not going to breed those anymore. So how would he go about doing that? Can you imagine how hard this would be? How much communication would be necessary in order for Mike to talk to a dog long enough for it to be distracted enough from its guard duties or from its food bowl or from its tennis ball in order to comprehend the idea that, wait, what are you saying? You want to tell other dogs to only have puppies with other dogs that are smart because you want puppies generations from now to actually own the humans? Wow, that's a cool idea. But how the hell would you ever do that? Well, the nice thing about it is humans don't have that problem. We're not trying to escape servitude. We're actually just trying to get better. And I think it's one of the most important things that I can start you with is that at the turn of the century, around 1900, most of the smart people in America and in the rest of whatever, I don't know if you can call it the first world in 1900, but let's say the developing world or, I don't know, whatever world came from Europe. The assumption was that. The vast majority of people understood this. And the higher you were on the hierarchy, in other words, the richer you were, the more likely you were to think this. Does that seem so crazy? If you think about it from the perspective of, if you can think like Mike, because, right, if you're just a regular dog who keeps constantly get distracted by the treats and the tennis ball and the smell and the girl and the guy that just walked past, and then you're never, ever going to be able to implement this plan because you only got like 5 years, because then you're not going to be able to find a female dog anymore. You only got like 5 years to talk to those puppies long enough so that the puppies grow up to understand that the rest of the dogs don't get it. You see how this time frame works? It's almost, you can imagine that if you don't start with a puppy and raise that puppy to understand that, we have to be smarter than the other dogs. Otherwise, we're just going to breed with the dog that the people give us. We're going to have to eat the food that the people give us. But I've got a vision, and I got to give this vision to my kids and my grandkids, and I got to give it to any dog around me that gets it. Now, I know that this is hard in some senses because I'm talking about a dog, but what I need you to feel is that if your parents were involved in the governance of America, then you were a possible candidate to be one of those puppies that got read in a little bit early that understood that not everybody knows this. Not everybody knows that we need to simplify the story for a lot of these dogs. You can't have puppies because, whatever. It's too many dogs. Oh my gosh, we're going to put, The Price is Right is going to tell you that you should spay and neuter your dog because there's too many dogs. And so that could have been the plan of Mike as well. Mike maybe got that advertisement on there so that only the dogs that he wants to breed are breeding. So what I'm trying to get you into the mindset of is to try to formulate what it would be in 1900 or so. I didn't bring the oldest of old books. Multi-generational ideas. Yes, multi-generational. Because if you're going to come up with a plan like that, it's got to also have a vision like that. And I'm going to try to argue to you with a little bit of discussion that you will find that this biology that we have right now is actually a plan or a description that are in books that are too old to have been predicting. It's not possible. You can just pass this one around. Again, I'm just taking a huge risk here because there's only us, but understand that these books could be anywhere from $100 to $500 each. So if you don't have clean hands, don't do too much paging. What I want you to do, though, and what I'm going to try to expose you to is the idea that textbooks don't do this. Textbooks are a summary that was put together by someone with ulterior motives. I'm not saying all textbooks are wrong. I'm saying that all summaries are probably wrong. Cliff notes are never good. And so you got to go all the way back to when people were first writing biology books to understand that not a lot has changed. So I'm just going to give this one over here first, just so you see who this is. This is Julian Huxley and H.G. Wells writing a biology book. The main message of this biology book is all life is energy. That's kind of weird, isn't it? Just page through that. It's a really cool book. Very hard to get. Another sort of, where did the little one go? Another sort of, is Kira there? Oh yeah, here. Another sort of inspiration for you to think about the turn of the century is that this is a Bible that was carried over from the Netherlands. It's only in Dutch. It's only got the New Testament of Jesus Christ in it. But this is the kind of book that somebody would have brought over on a boat from the Netherlands, just to kind of get you into the time period. And then this is actually really cool. This is the New Testament in Choctaw. I don't think they print these anymore. So just think about, again, what America is it where people are carrying Bibles over here, coming over to make a new life. They're translating the words of Jesus Christ into Choctaw. Think about that for a second. As you try to think about where we are. And then I'm going to pass around a bunch of books. You don't have to, look at all of them. You can look at as many as you want, take as long as you want to. These, I think, are especially interesting to pass around and look because you've never had this subject before. So in early 1924 through 1940, everybody would have had a course in bacteriology, which later became microbiology, which they don't teach anymore. If you get a microbiology class, it'll probably not be what this was. Notice that this book is called Bacteriophage Lambda. That's one bacterial virus with an entire book about it, okay? Just passing these around. Again, these are just examples of bacteriology books and how, it's not a small book. This is a course, probably 2 courses that were taught in medical school. There's no virology books from this age except for the ones I'll pass around next, and they're not medical textbooks. So why is that? Why is the virology book from this time period? Here's another one, just called Bacteriophages. Here's another one, Bacteria and Their Viruses. How is it possible that one of the only contemporary books of virology is this one? This is virology with a little pretty picture of a freaking plant in the front because, it came from tobacco or something like that. Oh, I'm not going to find it now. What's going on? It's like one very pretty plate. See? That's it. Tobacco virus. This is the virus book that is the contemporary of these bacteriology textbooks. Now I want, oh, sorry, just a marker. Oh, I probably lost this spot, but that's all right. The point that I'm trying to make here, again, is that there is a transition that occurred in biology where bacteria, microbiology, all the infections that happened in World War I were bacterial. All the deaths that occurred in World War I were bacterial pneumonia, bacterial infections of wounds. There's no viruses. They're not talking about viruses. How is it possible that these people in 1900 had one silly book about viruses, but they didn't die from them? Had all this bacteria stuff that we don't have to worry about anymore. The reason why I'm afraid is because a shift happened where, here's another really interesting book. I should check which one is the first edition. Second, this is the chewed up one. The chewed up one is the first one, I think. I don't know if these people on Zoom are not going to be happy with me, I don't think. That's all right. Oh, this is the wrong book. This is another good one just to pass around. Again, I'll just start it over here. What you can do also, if it has an index, go ahead and look at something that you think you might want to think about and see what it says in that book. Because in order to prepare for this course, that's all I've really been doing is reading old books to try and understand one thing. When did we decide that bacteria doesn't matter? When did we suddenly decide that immunity is basically antibodies against viral proteins? Because there's a whole book about just one bacterial virus, and all of the molecular biologists at that time, and incidentally now, agree that bacteria are plentiful. Where did my family go? Because that car is open. It's my car. Sorry, I just got messed up. What's that? Well, you could just shut the door if you don't mind. Thank you. What did I just say there? Sorry, that really threw me off. Oh, yeah. These books are similar in the animal biology range. These are also written by interesting people, Haldane and Huxley. This is Julian Huxley again. And Haldane is actually an Englishman who moved to India when he was pretty young, and he has a couple, hey, nice work. That's why the car's open. Rick, it's all right. So you can take a look at these two. And so if we were to just, that's the one thing that I find really annoying, because I wanted to be able to share like whenever I write things down with you guys, but I don't know. Oh yeah, you're doing that. Good. Clip it to the table. I'll stay out here until we get it clipped to the table. So my crew has come back with a monitor so that you won't be able to see it very well, but at least you'll be able to see something. So what I think we have to start with is the early 1900s. I think we have to start when we started to think we knew what we were talking about. It's World War I. What at that time already? What's the most important working assumption of biology at that time already in 1900?
Can I read this quote?
Yes, please go ahead.
John Muir.
Yeah.
It's when we try to pick out anything by itself, we find it hitched to everything else in the universe.
Very nice.
Yeah.
The books are full of those things.
Yeah.
If you take what Jay said about the dog, about the thing, multi-generational plan, the dog did. Teach their kids to have smarter dogs, teach their kids to have smarter dogs, and so on and so forth. Eventually, the parent dogs, they're long gone for decades.
It might be too big. We might have to.
Way into the dogs to know that the plan was ever made for a whole book. And if you go on the internet today or CNN or InfoWars, whatever is going on, left, right, you choose, you can be encouraged to think that Bezos, Elon Musk, and Peter Thiel, whoever, are some kind of masterminds that are trying to get you on a journey But these are just dudes who happen to come into the world
Unscrewed it.
following the plan. That is in these books. These books lay the foundation. I swear to God, you cannot understand anything going on today without realizing that these books lay that foundation down. You just have to view it through a little bit of an interpreter. Jason can help you.
We're still going.
I've read the whole book.
What's the working assumption that I'm trying to get to? Somebody can answer. The biggest working assumption of all. Galapagos Islands. Evolution, right? The biggest working assumption of all is evolution. That is a story that's very old, and a lot of people understand it, right? In some ways. What are you doing?
I can't go on there. The table's too thick for that one.
Oh, okay. Well, then let's do something else with this. We're not going to worry about that now. This is why what we're dealing with here. This is why it's real. So evolution is the probably primary working assumption of modern biology. How far does that go? I have the word of a Nobel Prize winner named Edward Moser, who I worked with in Norway while I was still an academic. And he has an interview that he does in Norwegian, but I think he actually speaks in English. And he explains modern biology and how he thinks about it. How far, I'm trying to ask the right question so that you will answer it, but how far exactly does evolution go? Ultimately, there is a book by this guy, Haldane, who wrote that biology book with Julian Huxley, but also he wrote this little book called Daldi Adadios in the Future. This guy had the right idea in terms of what evolution is. And in this book, and in what I would like you to think about, evolution is the ultimate in materialism. It's okay, technology is good in the sense of that's maybe the ultimate expression of it, but where I'm going again is for the root of the working assumption. The working assumption is materialistic, all the way down to the roots. What does that mean to you? I'm trying to use big words that other people will use against you and try to confuse you. So what does it mean when I say that evolution is the ultimate materialistic theory? That's for sure, but then answer my question. What do I mean when I say that evolution is the ultimate and materialistic thing? Can you tell me what isn't true? For sure, if you believe in evolution, what's not true? God? Do you have a soul? No, of course you don't have a soul. And that's what I mean. Edward Moser, Nobel Prize winner, interviewed about biology. Ultimately described, because he's a brain scientist. Tomorrow we're going to talk a lot about my favorite subject. And he's a brain scientist. Asked, what do you think about a human and consciousness? He said, well, there's no soul. It is all a physiochemical process that gives the illusion of free will.
Like we'll call it psychiatry, but the psyche's got nothing to do with it.
Exactly. But you have to understand whether you accept it or not, the foundation of modern biology is ultimate materialism. It is dirt, it is chemistry, it is physics, but there is no spirit, there is no soul, there's none of this stuff in biology. Now, you can learn biology from people who believe in that stuff. You can go to DePaul University, learn from people that are Catholic, and they will tell you that they believe. This is the most important message I've got for you all weekend. After this, you can go home. What do they tell you? If you go to DePaul University, you interview or you talk to, you're thinking about going to DePaul University, you sit down and this guy is interviewing you and you're thinking about being PubMed at DePaul University. That's where I went. And it's a Catholic school. What do they say? Do they believe in God? Of course they do. They're Catholic. So how do they reconcile these two things? Can you explain it? Just what is the way that they make these two things fit together? It's really easy.
Doctrine.
Huh?
A thing called doctrine.
Doctrine. Okay, it's even simpler than that. I really want to get down to actually what these people do in their head, because I was doing it for my whole career. Oh, that's close. Not really, but that's exactly the wrong words, and it's perfect you said that. Intelligent design is what she said. Intelligent design is what? That's like a creator that puts something together. It's like a watch. That's okay, but that doesn't work for a Catholic biologist who's got to teach you biology, who's got to teach you evolution. So what's the answer? How does a Catholic biologist get away with teaching biology where there's no frickin' soul? Do I have to fill it in? It's because he just says that God did it that way. God made us evolve.
That's what I meant by doctrine. You don't
There you go.
let them in your mind. You just
Then Rick is saying it without saying it, and that's why I'm the teacher and he's the student, but he's right. Is that it's a doctrine. They just accept the idea that biology can exist and this little doctrine over here exists, but in reality I understand that as a biologist, this is just a doctrine. I'm just a Catholic guy. None of those people are wearing a scapular. None of those people are going to frickin' Catholic confession. But they are willing to say they are Catholic, and they are willing to say that they teach biology at a Catholic school, knowing that they are teaching pure materialism. There is no soul. You come from the dirt, and the dirt you will return kind of sort of sounds like that, but it's not. They don't even know that they're hypocrites, though. Why? Why don't they know? They don't know because how do they know how God did it? Why couldn't God have used 6 million years? Why couldn't God have made the dinosaurs and said, that sucks? Why not?
I don't know.
But you know what's so magical about that is that means that everybody accepts evolution. It's just a question of whether you put the ultimate cause at some old man with a lightning bolt in his finger, or whether it's just some lightning bolt in a mud puddle. And that's a pretty magical trap that makes everybody accept the same picture of evolution. Do you see how crucial this is, sir?
Be clear, you're not saying dinosaurs.
I'm not saying that at all. I don't have to. No, I don't have to. I don't need to say that.
There are Catholics who don't believe in evolution.
Yeah, that's true, but then they're not teaching biology. And I was specifically, let's say, indoctrinated at a Catholic university by Catholic people who were able to explain to me very simply, you can accept evolution and Christ because Christ did it that way.
So that's wrong.
Well, I don't know whether it's wrong, but somehow or another, if you're going to be an academic biologist, you're going to have to reconcile this with that at a Catholic university. And so that's why I am very familiar with the way that these people straddle those two. They introduce themselves like that. Are you kidding me? I was an altar boy, when I was young, and now I'm a biologist. I like to study diatoms, and, I'm really interested in the marine life, and the only place I could find to work was here. That's how it goes. So these people, by their own practices and by the own thing they do, they don't actually need to verify that every day. Not every day are they asked, so you still no soul? You still sure about that? That's not how that works, right? It's just a kind of thing that you put in the back of your mind. It's a working assumption. I can accept evolution because all these people say there's all this. I'm not suggesting to you that there's no evolution. I'm suggesting to you that there could be evolution, and it could happen on different time scales. I'm telling you it doesn't really matter because people are governing you for different reasons. And they're using that as a thing to make you surrender to the idea that you're just a blip in time, one of 6 million odd years on Earth. Well, guess what? The only people who are alive arrive right now. And if you don't do anything now to pass on to the children, if you don't plant any trees under whose shade you won't shelter, then no one will know you. That's for sure. So I got a little sidetracked there for a second. I would like you to take about 5 minutes to, take a little bit of a break and give me a second to kind of reorient where I want to go from here. And we have established one thing about the working assumption is evolution. Now, what I would like you to do is try to come up with, now we already said the one big one, which is that there's no soul. But now I would like you to fill in as best as possible for me on a piece of paper or somewhere that I can come around and look at a little cartoon to try to describe to me what you think oh yeah, perfect. What you think if you had to draw a cartoon of evolution and you were to put bacteria and fungi and animals and reptiles and whatever and birds, just make something that, whether you believe God did it or whether it's a lightning bolt in a mud puddle doesn't really matter, but just so that I can get an idea of how you would draw the tree. And then also we can have something over there. And can I send them over now? I mean, what are we doing? Because we're already behind time, right? Or what? Yeah, that's what I mean. So this is a class. This is not a show. So the class is going to keep going, and the Zoom meeting is going to keep going, and they can keep tapping away. And Kira will check every once in a while now that he's back to see if anybody's complaining or whatever. And so please don't feel, I know some people well. I know some of you guys not at all. So when you want to say hello, say hello. If you don't feel like saying hello, that's also okay. But I love everybody. I love everybody on Zoom. But primarily I'm here for you guys right now. And so that's where I'm focused. Again, if you get your fingers dirty, don't forget to get your fingers clean again if you want to look at some of these books. I don't have a way of really showing you a good picture, so the only thing I can really do is draw it and then maybe carry it around a little bit. So first of all, just again, take a couple seconds to make a small square on your piece of paper and try to say with a cartoon, how did life evolve? And I will draw one too. How about that? I will do one also. Now quit switching between those two. There we go. So I would draw a cartoon or a tree starts somewhere here. Here's the lightning bolt. Doesn't have to be God. It could also just be a lightning bolt in a mud puddle. And then somewhere we get life to come out, right? No, it won't work. There's no projector, unfortunately. They said there was, but there isn't. I'm going to have to carry it around. And so the idea would be time on the axis down here on the bottom. I'm talking more to the Zoom now. And then here maybe would be, I don't know, diversity or cell number. And so as we got more complicated, and then maybe there's this and there's this, whatever. And so I'm just going to carry this around the room and show everybody what I mean and what you might have come up with. You don't necessarily have to have an X and Y axis. And again, I apologize. This is very crappy instruction. I should have a beautiful whiteboard or something like that, but we'll get it right next time. So the idea is that somewhere here in the bottom, you're going to start with that lightning bolt in a mud puddle and then life diverges away from a single point in that mud puddle. And you can have in this early part of the evolutionary tree where there's a mud puddle and then, bacteria and fungi and all these things spread out, somewhere, I guess, fish turned into amphibians, then amphibians turned into reptiles. Reptiles were briefly ruling the world as dinosaurs, then died out, and we're still left with crocodiles, and now we have ourselves here because we have technology and that's why we won, or something like that, right? But at the end of this graph is where we are. Now, as far as evolutionary theory goes, from the beginning till now, how much do we imagine bacteria have changed? Actually, not at all, I think is the right answer. They haven't changed at all. In this entire graph, there should be a line that comes out of the mud puddle and then just goes straight to modern time. They didn't change at all, according to the way this works. They were bacteria. And then, if you take these books on the table that are here next to Janet and pass them around, you will see that there are some theories that some of the organelles in cells are actually organelles that were bacteria before they came organelles, particularly the mitochondria. This is a book by a person by the name of Lynn Margulis. And if I can just take a brief sidestep here, this book, written by this woman who doesn't cite Joshua Lederberg very often, but Joshua Lederberg actually writes the foreword to her book. And the reason why is because Joshua Lederberg, in my humble opinion, being one of the more important biologists, maybe the most important biologist in America you've never heard of, the inspiration for this course, the inspiration for my friendship with Mark, and the inspiration for me thinking that there's something for me to do here for the rest of my life is Joshua Lederberg. I think I would have been friends with him. I think I would have been his favorite student besides Lynn. Now, the thing that's cool about Josh, and this is another great example of this book here, is Josh is not like the people that you find in academia now who stand in front of their students. Essentially, I have a postdoc who did this, but it was my idea. Josh is very good at letting his students take his ideas. I'm pretty convinced that almost everybody that went through Josh's lab came up with good ideas because they went through Josh's lab. And had Lynn Margulis not had him as a teacher, Lynn would not have come up with this idea. Had Samuel Epstein not had him as a teacher, Samuel Epstein would have never led multiple NIH conferences about how LSD and other drugs can change gene expression permanently. I don't know why we don't have this in our basic biology books, but actually pesticides, chemicals that you take as medicine, even shitty food, can change the expression of genes in the bacteria in your gut and also in your own cells. And that gene expression change can be permanent. It can be irreversible because you are on a trajectory through time. You need to think of yourself as an arrow already in flight. If you think of my son as an arrow that I can pull back, but once I let go of the string, that's where he's going. And after that, if I want to adjust his trajectory, if you think about it like a boulder going down a hill, or you think of an arrow on the way to the target, it's kind of late. And the best thing you could do maybe is nudge it a little bit. You're not going to get it to go in a different direction anymore. And so from the perspective of students, I've found so many examples of where Lederberg actually just kind of disappears. It's got to be his idea because he's behind him, but he disappears. I'm not saying Lynn's not smart. I'm saying that behind all of these most inspirational ideas I've found are Joshua Lederberg. So again, we're still trying to work on this assumption, this working assumption here on this thing. Now, the next thing I want you to do is remember that biologists love probabilities. So what is the probability, for example, that for 6 million years after the creation of, or the spontaneous generation of bacteria, that bacteria didn't change? I thought the whole principle of life was changing, constant evolution. Yet we have at least one thing that all biologists since the 1920s agree hasn't changed. In fact, Joshua Lederberg will tell you that everything we know about DNA is from bacteria. How was the COVID-19 vaccine made? Using bacteria. In fact, the only thing that you can know for sure is that until we could make large quantities of DNA, we really couldn't study it. And so bacteriology is where that came from. Understanding how bacteria juggle pieces of DNA is where that came from. And in fact, if we think about these working assumptions here, what's involved in making this tree is some kind of systematic, millions of years of improbable changes. If we believe evolution as it is, and again, I'm not telling you that evolution didn't happen, I'm telling you that evolution, this is for Zoom at 2, that evolution as an idea, which is acidic and dissolves all other ideas, is a falsehood. The fact that birds can change, that birdsong changes, that's all great and that's all wonderful, but the idea that we came from nothing is actually the ultimate materialism of evolution that we don't necessarily need to teach while still acknowledging this diversity around us. The fact that from the time when you're a kid to the time when you get old, the butterflies might change. The cardinal song might change. It might even invert. But that's over time, not the same kind of change as what is portrayed here, which is again, many, many, many improbable events for many, many thousands of years, which accidentally, if it went wrong, we would still have velociraptors and they would be running the thing. That's the part about evolution that you got to get your head around, because the only thing that we know as biologists is what we know now. And what we know now is that the world is completely dominated by bacteria. Absolutely and totally dominated by bacteria.
They're certainly critical to my life form.
Of course they are. They're critical to every life form. We can go into, for example, bees, and you will find that they have compartmentalized stomachs, and in those compartmentalized stomachs, they have different bacteria in each compartment, the same way that a freaking cow does, and the same reason why you have 22 feet of intestine, because the microbiome was not the same at the start as it is at the finish. It's the same reason why you have Peyer's patches in your intestine, which allow the bacteria to interact with your entire lymphatic system at night when you sleep. So what I'm trying to get your head around is the idea there might be, if you think of all of these branches in this imaginary thing, right, that's going to lead to, right, don't forget what it leads to. Where's one of those little blue books? It leads to the Haldane book, right? It leads to animal biology. And let's just stick with animal biology. How many reptiles can you name? How many birds can you name in Pennsylvania or in the eastern side of the United States versus western side of the United States? Have you ever watched a movie about people who try to find the most birds in a year? There is some pretty spectacular biology out there that is still all explained by non-likely events. Very non-likely events over many, many thousands of years. Now, what I want you to understand, we'll go back to where we were before, everybody accepts it. Even the person who says God did it that way is not sophisticated enough to say, why would God do it this way? Because remember, we're almost there. This Lynn Margulis, I think, was almost there. She sees that mitochondria, the powerhouses of our cells that you only get from your mom, is probably the reason why you should keep extra track of the genealogy of your moms, because your mitochondria are ultimately from them. You didn't get any mitochondria from your dad. It almost makes sense why the Jews keep track of Jewishness through the mom only, because that inheritance is different than the combined inheritance. Yes, sir.
The benefit of mitochondria is?
Oh, sorry. It's this little tiny organelle in the cell that does all of the ATP production. You have thousands of them in your cells. They have their own DNA. They express their own proteins because they have their own ribosomes, which make protein. And so basically, they are bacteria, but they're stuck in your cells for some reason. We don't talk much about them. Now the health freedom movement says that mitochondria are important, but they don't really tell you what that means. But the really interesting thing to think about here, and I have to say that there's one thing that I'm not disappointed in, but I guess I have to be very, I kind of imagined that everybody would have a laptop and then I could say like, open this PDF, and now that can't happen. But in your PDF group there is a paper from 2017 where the first people ever proposed the idea that bacterial viruses, which are called phages, actually also interact with our cells. Before that, no one had ever assumed this. No one had ever proposed this. So now remember, again, this is so hard for me to do without a screen. It's yes, go ahead.
All the cells in your body may have sub-organisms within them, thousands each, which are in reality potentially remnants of other individual bacteria.
Yes, individual bacteria.
Have their own DNA.
Yes.
Inherited from the mom.
Their own DNA, their own ribosomes, which means they don't need your cells like a virus to hijack your cell machinery to make their frickin' proteins. They don't need to do that. And so now what I need you to get your head around is the idea that in early 1900s, the only way to cure an uncurable bacteria infection was to use a phage. And the French were very good at it. The Russians knew all about it. They understood that you could find a bacterial virus, which is called a bacteriophage, that would end a bacterial infection that you couldn't end any other way, and end it completely. And the way that they figured this out, which is very interesting, is in our favorite place, India. This guy from France, whose name is going to escape me, but I'll bring it tomorrow. It starts with an H, and I can see it. It's like HEU, something, LLES, something like that. He did a lot of research on phages, and he went to India, and what he discovered was that when you went to the Ganges, you could find places where cholera was breaking out, and then you could go downstream and there would be no cholera. And he started to do research on what was in this water and what was in that water, and he started to be able to indirectly, because this is like 1890 or 1900, they're not doing this with PCR or other bullshit that they're doing now, claiming that they're doing high fidelity stuff. He could only do it by showing that a bacterial culture that was cloudy, it's one or the other, then if he put this phage in there, it would become clear, or vice versa. Whatever the read was, if you had this really nasty bacteria growing in here, let's just say it's cloudy, and then he could add a drop of this stuff from the other part of the Ganges and put it in there, and then it would go clear. So all the cells would rupture, and they wouldn't stop the light anymore, and so then it would become clear. And so he had figured out that there was some signal that was circulating through the Ganges, and if you went to a place where there was no cholera, you could take that water and put it where it was, and then the cholera would get just wiped out. And so he called that a bacteriophage. And he is a very famous guy operating in France around the same time that Louis Pasteur is operating in France. Louis Pasteur is one of these people who, of course, is the germ guy and the pasteurization guy. And so understanding that this is happening in 1920, at the same time that Julian Huxley and his dad and H.G. Wells are writing a book about biology and books about biology and telling us that, what's the famous guy who discovered you could breed plants and then they would segregate stuff? Because Mendel's old. Mendel's an old monk from an long time ago. There's one thing you might not know about Mendel, is that he first spent many years breeding pea plants that bred true. So in other words, he started by creating purebred dogs, and then after he had purebred dogs, he crossed them, and surprise, surprise, the babies had two primary outcomes. It's not that dissimilar to the goofy stuff they're doing now with doodles. So you take a poodle and a Labrador and you put them together, and basically you get one outcome. And if you don't get one outcome, we get one of two outcomes. He's either more poodle or more Labrador. And so if you do that with pea plants, and you're only looking at wrinkled seeds or whatever he looked at, then you can get this phenomenon very similar to, I think, the same phenomena that they will sell in the Human Genome Project, that there are exceptions to a rule, where you can make it seem as though, wow, this is pretty simple. I guess this is how it all works. And that only happened because he spent so much time breeding those plants. But now remember, that's before 1900. So we're going in to the early 1900s, to bacteria, to not knowing how heredity works, and the vast majority of the world actually thinks that proteins do it. Most people are pretty sure that proteins carry most of the information, they do all the work. So the only reason why they thought it might be nucleic acids is because they were finding nucleic acids in the pus of people with pneumonia. And so they were just a little bit interested, because what I told you before, World War I is basically all bacterial diseases, all bacterial deaths, and the worst one was pneumonia. And so there's been a lot of focus on pneumonia. You might even be familiar with the idea that that's one of the worst, or not one of the worst, one of the most common ways for old people to die to this day is bacterial pneumonia. One of the reasons why, and one of the things that these COVID people are allowed to say is that, they didn't give them antibiotics and so they divide, but they don't actually get all the way to where we're going, but this is part of this. So antibiotics now are of course on TV and people are saying we shouldn't use too many of them because we're going to have antibiotic resistance bacteria, but there's a guy in Pittsburgh who drives around in a car that says phages on the back of he's a Indian guy. It's a BMW. I've stopped him in the street and told him, what the hell's going on with your license plate? And he actually said that he does phages. He consults with people who have chronic infections and this other shit, and he helps people all around the United States find a phage that gets rid of this chronic infection in their joints from an operating room. Persistent stuff from, the bacteria that are found in hospitals are sometimes especially nasty. And so this guy just exists. But they exist in the background. And the background that I'm trying to point out to you is that these books exist, that there is a bacteriology. And each one of these books is a testament to the idea that something was lost. Something got lost because this guy, Joshua Lederberg, and because I can't tell you to pull it up right now, I won't tell you to pull it up right now, Joshua Lederberg wrote a very wonderful essay. And he's written many wonderful essays, and many of them are included in that PDF collection, where I could put it up on the screen too, that he describes the impact of discovering finally that DNA describes what Mendel did. Because remember, at this time when Joshua Lederberg gets his Nobel Prize when he's 30 years old, again, remember, you haven't heard of this guy, but he got a Nobel Prize when he was 30 years old. He was an advisor to John F. Kennedy when he was like 31. So it's strange that I'd never heard of him either until Mark Kulak told me about him in 2022 or 21. Joshua Lederberg got the Nobel Prize for contributing to our understanding that DNA is the big deal, but he actually didn't do the experiments that he would say are the most important ones. There's a couple really important ones that you can look up and read, and I think you should master these experiments. There's one that's called the Hershey-Chase experiment, where they use bacteria and bacteriophages and they label the protein and they label the DNA with different radioactive elements, and when they observe what happens when they expose these bacteria to the phage, they see that the labeled protein of the virus stays outside and the DNA goes in. And so this was the first proof that the DNA was carrying the information and not protein. And a lot of people's, they were betting their career that was wrong. And then that experiment came out and it changed a lot of things. And Joshua Lederberg and a couple other people like Rene Delbecco and this guy, I think, and this guy, Max Delbruck, were right in the position to hammer on this. So and you have to see this. In 1930, they're teaching bacteriology, and they don't know that DNA does it.
That's from 1962, Watson and Crick. Did they ever heard of
It's 55 or something, yes. So what I'm trying to say is that all those bacteria books that already accept evolution, already accept that it's just a matter of time before we figure out all the steps between bacteria and ourselves. They already know that bacteria have viruses that they don't really understand, but use DNA to kind of wipe out bacteria, but then they come back. And so it's a little like if bacteriophages worked like eliminating, then there would never be any cholera in India anymore, right? The phages would just wipe it out. But it's more like the leaves come on the trees over here, and then the leaves come on the trees over here. And so you can start to imagine a scenario where we're not dealing with the kind of biology, the kind of discontinuum that they would like you to believe. We're dealing with something a lot more cyclic and a lot more dependent on this cycle, which is bacteria and their bacteriophages, which we do not talk about at all. Now, again, I'm jumping around a lot here, but I just mentioned that in your little folder, there is a article about bacteriophages infecting, bacteriophages becoming pathogens of or interacting with our own cells. Now, understand this without a doubt. Right now, and from 1920, the assumption is evolution. The assumption is we don't have a soul. We still have that same assumption because I worked for a Nobel Prize winner who says that. We also have left bacteria behind. And you know this in your heart. You know this from your experience of the last 6 years, that bacteria played no role in the biology of the pandemic. Not one. Not one bacteria was mentioned. And actually, the first thing you've heard since a virus is a protozoa. Cyclospora is a protozoan. So it is spectacular to think that a medical school course called biology was called bacteriology was taught for 50 years in America. A Nobel Prize winner at the age of 30 came out of bacteriology. And what we believe we know about this answer is all based on bacterial genetics, because bacteria haven't changed since the beginning. And so the only thing we know is bacteria use DNA to send signals to each other to cause a bacteria to lyse or to explode. They can trade genes, and that's what Joshua Lederberg was actually got the Nobel Prize for, because he showed that bacteria can connect with each other and then pass circular chromosomes to one another, so that traits like, for example, resistance to an antibiotic or maybe even enzymatic function could be passed from one bacteria to another, and that was also carried by DNA. He would not have been able to answer that question had they not done the Hershey-Chase experiment a few years earlier. So this was a very rapid time, but notice that the working assumptions are not changing. The working assumptions are not changing. They are fitting their observations in bacteria to explain the entire tree. I guess we're right about everything because DNA does it in bacteria. Now, I'm not suggesting to you that there aren't chromosomes and that chromosomes aren't doing. Yes, of course. I believe this. I do. I think that biology in some miraculous way is encoded in the smallest molecules of DNA in mitochondria, in your mom's egg and your dad's sperm. I'm not arguing about that. I don't know exactly how you became who you are, but I'm absolutely certain that you guys wouldn't be you without him and her. That's it. And that will never happen again. Do you hear me? It'll never happen again. It'll never happen again.
Thank God there's only one of me and you.
Yeah, but, and now I'm going to try and make a jump, and then I'm going to take another break because I'm feeling a little need. It'll never happen again. And this is crucial because the Human Genome Project isn't done. And I'm kind of talking to these two young guys because I know you guys know this already. But you're familiar with chess. You're familiar with this, I think it's a Japanese game, Go. It's got black stones and white stones. And it's a big table, and it's got like a grid, and you just put white stones, and if you trap the other guy's colors, then you get to flip their stones, and whoever ends with the most stones wins. So chess is a very complicated game. It has a certain number of outcomes, but you can also imagine there's a certain space. If we were to play chess and you were good, that I would make really dumb moves, and then those are kind of not really part of the game. And so if you were going to try to solve chess, you wouldn't worry about those. And Go is kind of like that, but it's a lot more possibilities. And so I hope you're familiar with the idea that they solved chess. They beat the blue brain computer, beat a human, or beat Kasparov or whatever, and then finally the mainframe computer wins, right? Computer finally beat chess. And then
You cannot master Go. Is
No, they did again. Then like 5 or 8 years later, Google did this one because it's so many more possibilities. And for a brief period of time, they had declared victory. But then a really smart Go guy did what I just suggested to you. What if I do dumb moves first? And then the machine learning algorithm didn't know what to do with the dumb moves. And so then for a little while, the human was able to beat him. But then after those games and that experience, the machine learning algorithm plugged those holes and now they can kind of win. At the same time, I want you to understand that in 2005, Tony Fauci and Francis Collins and the entire National Institutes of Health announced that the Human Genome Project was done. Now, well, I just want these guys to get it and you guys to get it, that's a lie, but it's a lie of a national security type lie. Because the only way that anyone is ever going to crack the genome using computers is if they have every one of the games. You need all the chess games. You need all the Go games. And you need all the human games. You cannot go to India and sequence all the Indians and know anything about you guys. Nothing. You can make a map, like if you think of a national park, it's got an entrance, it's got some campgrounds, it might have some game lands, and it might have a shooting range. And you could have a general map of a national park, but you don't know what that national park looks like. And what I need you as a young person to understand, because these adults are too old to do anything about it, is that they need you to accept yourself as an experimental animal before they can monitor you enough to get the data they need. It's not a game. With chess, they have a game, and the game has rules, and at the end, there's a winner. How do I evaluate you as a game to put you into a machine learning algorithm that it can compare to your brother and compare to Mark and to Janet and then get a win? Because that idea is from frickin' Joshua Lederberg. This 30-year-old guy, microbiologist, bacteriologist, already a long time ago, that we could bankrupt our entire civilization if we tried to use computers to solve the puzzle of evolution, the genome. Remember, it's a roughly 3 billion base pairs, and the only thing we know is that some of this DNA codes for a protein. Everything else we just assume. If you put all the right proteins together, I guess they just assemble and they're you. If we put a few more proteins in together, I fall in love with my wife and have kids, and that's it. Because they have no other explanation, because it's all materialist. It's all materialist. There's nothing else, and more importantly, it has to come from one point. Now, these people over here, the symbiosis people, don't even see it. But if you put the complication in the middle, where the bacteria are a part of us, the genome can code for a lot of things, because that's what Joshua Lederberg's biggest message was. Right now, evolution teaches you that you're your genes. You're screwed. That's all you get. Joshua Lederberg's most important message was that's half the puzzle. Eugenics is the Nazi part. Euphenics is the lifting weights, eating good food, reading the right books, practicing every day, the discipline that's necessary to get skills that you cannot be born with. You can have the potential to be a piano player. If nobody puts your hands on a piano and teaches you what it is, you're not going to be a piano player. And the most important thing to understand with evolution and high school biology is that we are teaching our children that they are their genes. Rather than teaching them that the genes are kind of a unknown potential on which you can maximize, we just kind of pin them back in this idea that you're stuck with this. And now the trick here is to see, and I've said this before, earlier, that creationists believe the same thing. They just explain it with God. God did it with 6 million years of evolution. And the evolutionary people just say you don't need God, you just need chemistry and physics. Now, the reason why I think this is contendable is because of these people who talk about symbiosis, who talk about asking the question, why do we have all these things that look like bacteria in every cell of our body? The more of them you have, the better off you are. They can measure it in people. If you're a martial artist who's trained for 20 years and they measure the mitochondria in your muscles, it'll be over the charts compared to you. Because after that training, all those years, not only is their myelin better to connect those muscles, to make them spike and to fire together, but they actually have more mitochondria. Mitochondria divide on their own. Do we have a drug that makes that happen? How come we don't know anything about that? Wait, what, they divide on their own? Wait, what? But how else do we get mitochondria in every one of our cells by the thousands and start from one egg? So somewhere they're not organelles. They can't be organelles if they start out in an egg and they're not the only ones you have. All your mitochondria are not in an egg and then you spread them all out through the 3 trillion cells of your body. That would be preposterous. So throughout development, you have bacteria that are dividing in your cells as your cells grow. It's not explained in any biology book in existence. Nobody gives a shit. And if they do, they just say, well, they must have just kind of, got together in a mud puddle. And again, what is this? It's still this improbable thing. Watch how easy it is. I don't know if I can do it to make the impression that I wanted to make, but I'm going to try anyway. What happens if I start with a genome that can make 4 legs and a head, and I start with and I start with a microbiome, and I start with cells that have mitochondria, and then I say, go. Now we get all the world covered in bacteria. We get the whole world covered in bacteriophages. We have a one kind of prototype genome that can produce 4 legs and eyes and a head, and you have almost no variation in that. It's always 4 legs and a head. You have 4 legs and a head. Hate to break it to you. And so what's cool about that is look what happens. If you start here, then you can have one line to bacteria. You can lose some of this stuff and you can make it fungi. You can lose some of it and make it archaeobacteria. And you can use development and environmental pressures to change the shape of the 4-legged head that you produce. And now all of a sudden, how many improbable events do you need? Just the one. Just the one where all that shit comes together and it works. And so now the watchmaker doesn't seem like so crazy because maybe bacteria is something that came from a star. Maybe archaeobacteria is something that came from another star. And it got on the earth and started in the middle rather than starting at the bottom of a tree and infinitely many improbable events that now lead to the diversity that we see. Where all the while through there, what's not changing? It's DNA to RNA to protein. And you need DNA to RNA to ribosomes to protein. It doesn't change at all. So how is it that we went from one point to many points, but certain things didn't change at all? That's so incredibly improbable. It's not because things that didn't have the central dogma died. It's because it started there. And if it starts there, you don't need to explain this huge tree with 6 million years of evolution from nothing. You can start in the beginning where everything is together. And then if you have about 10,000 years of things walking around taking a shit, then you have microbiome in the soil. You have all of this stuff happening in the water. You can even imagine a scenario where life went into the water instead of coming out of it. And so we all agree that that's what happened with whales. They went back into the water. Now, what I'm suggesting to you is not that evolution didn't happen. I'm suggesting to you that evolution plays a trick on you and that everybody plays the same trick, whether they're a creationist or whether they're a biologist. They accept that bullshit, improbable tree that explains everything that comes from one point. And that's not really likely. It's likely that that one point is in the middle. And if you thought that way from the very beginning, you might imagine evolution being a different process. Do I have that answer yet? No. That's part of the reason why you're here, to tell me that this is nonsense, to tell me it doesn't make sense. But at this stage, this is the kind of thing that we need to do to challenge this dogma of biology. I'm not saying that DNA to RNA to protein doesn't exist. I'm trying to challenge the idea that that supports the tree of life. Because I don't think it does. I think something like this supports a radiation of life. And if you thought that way, you might not need so many improbable events. And more importantly, you might come up with relationships that make more sense in terms of the microbiome and in terms of phages and in terms of our own mitochondria, which, if you just take that one leap, if the phages in your microbiome are able to pass into your lymph, which we all agree they can, then why couldn't those phages go to your mitochondria and talk to them? And do the same thing that those phages would do in a bacteria? Could be good signals. It could just be like, yeah, great, good to see you. It could be the symbiosis. And think about this one more thing before we break. What sits in between there is your immune system. The thing that determines how much access your bacteria on your skin and in your gut have to your insides is your immune system. Your immune system can decide if these bacteria can talk to your mitochondria or not. And why couldn't they? Why wouldn't they be? Why have we not considered this, given the fact that Lynn Margulis and these other people are writing a book about symbiosis? Then why don't we talk about the fact that that communication is available? That it may be even central to what we are, that our immune system is actually an interface, a molecular interface between our microbiome and ourselves, a permission to interact, rather than this, antagonistic relationship where we keep everything away. And I think that's kind of where I want to go after this talk of our friend Antonio. So I was just sharing with everybody, now I'm going to get hot again. Trying to come out of the dinner and try to finish up the Friday with a little bit of the working assumptions that we've come up with. And I've got here on the white piece of paper the assumptions that we came up with were evolution and the central dogma, which is well, come on, Jay, learn your buttons here. The central dogma, which is DNA to RNA to protein. We don't really mention ribosomes, but of course we should. We've mentioned the idea of genes going back to Mendel and it being more of a concept than an actual quantity. And talking about bacteria as the basis for a lot of the background noise of genetics, and that they have phages, and that this was a whole devotion in molecular biology, and in medical school there were whole textbooks about bacteriology. And we don't have bacteriology textbooks anymore, and we don't even really have microbiology textbooks anymore. We only have molecular biology textbooks, and that's a reason. The reason why is because they are oversimplifying an irreducible complexity to molecular biology. And what that does is it bridges this cognitive gap between the unexplainable and the explainable. How did we get here? It's just physics and chemistry. And that gap can be filled by chemistry and physics. That's really the end of it. So just to reemphasize what I just told everybody but without a mic, these are examples of street advertisements that would have been put in people's mailboxes in Paris. These are advertisements for bacteriophages being sold by the Professor D'Herelle. And I do not know how to say that name in French, but it's D'Herelle or something like that. And he's a very famous molecular biologist who is the discoverer of bacteriophages. But if you look into the history of bacteriophages, there's two discoverers, and they discovered them at the same time on opposite sides of the planet. And this guy, the French guy, made more claims than the other guy made, and so they discounted this guy's idea and not the other guy's idea. I don't have all the details on the top of my head right now, but I know that this guy got it right. And so at the time, in the same time that people were using hypodermic injection to apply combinations of substances, including opioids, under the skin, people were also in firm belief in some sects that bacteriophages were the way to cure infections. And a crucial piece to the puzzle of bacterial biology, but a very hard one to work with. That's why there are only a few people who do it. But these are real evidence that this biology is absolutely 100% real. And so, again, I'm trying to make this point. I tried to make it by letting the class in the room see the textbooks that I've collected. There are no shortage of textbooks that describe this field of biology, and they're just not used anymore. And I don't think that's because bacteria no longer plays a crucial role in our lives. I think that bacteria play a irreducibly complex role in our lives, and that's why they have to ignore it, or at least pretend that they're ignoring it. So one of the other things that we got to at the end with the help of Antonio is the idea of population pyramids and the idea of all-cause mortality, the idea of life expectancy. If life expectancy in a certain country is 75, then somebody who dies younger than 75 is living a shorter life than they expect. And population pyramids can allow governing people to make decisions decades in advance. We looked at a couple examples in the break of population pyramids that were obviously made up, but you could read things from the population pyramid, like who could find dates and who couldn't, or who would be available for jobs, this kind of thing. And so if we look at the bottom of the program, the main thing that I wanted to get through in terms of this course is that you would understand a little bit more about why RNA cannot pandemic, a little bit more about why intramuscular injection might be dumb, and population pyramids being real. The last one I think is already established. It's just a graph you've probably never seen before, and numbers you don't really think about, but they're actually absolutely essential, because in 100 years, the only people who are going to be here are the people who aren't yet born. And they pay very close attention who's being born, where they're being born, and where they're not being born, because that's all that matters. That's all the biology that matters. And so the final sort of conclusion that we've come to, or the place that we're at, is that the Human Genome Project as a biological endpoint could not possibly have been completed in 2003, 2005, or 2008, whenever Tony Fauci and Francis Collins acknowledged that it was done, or kind of done, or they did 3, or maybe they did 8, I don't know which. Craig Ventner is busy in the ocean collecting DNA. I don't know why he would possibly do that, but he doesn't think we need to do any more human genomes. He's in the ocean. So there's a lot of people who have been giving us clues to the fact that the Human Genome Project is not done. And in fact, I think the biggest clues were given to us by Joshua Lederberg, who I believe, thanks to the help of my friend Mark Kulak, I really believe that this was his idea. That he actually understood that it might be possible in a future where computers are bigger and the measurements are more precise and all of the people are medical experiments. But he did not imagine that we would be able to take over the evolution of the human race by any small measure, or by teaching every kid everything they needed to know about biology. And that's one of the reasons why I started the course with this idea of Mike the dog, because in order to get yourself into the proper mindset, you need to think about the problem of how do you get children on board with being experimental animals and not really, caring about sovereignty or personal rights or privacy or any of these things that we as adults seem to think are important. So biology, I believe, is the way that they're doing this, because biology at the state that it's in right now is absolute maximum materialism. And so if we're going to go all the way to making you into an experimental animal, you have to accept that role as an experimental animal, that you are just a cog in the machine. And so that's why evolution, that's why DNA to RNA to protein, that's why genes, that's why population pyramids in bacteria are not talked about, and the first three are all we talk about, because the working assumptions of modern biology are enslaving. The working assumptions of modern biology entrap you and make you assume that you don't have any control over the future of your family or any of the control over the future of who we are, when in fact you are, and our children are, the only future that we have. And so if this is the state that where we are, I'll turn around now. If this is the state where we are, we have these three things that I'm trying to get through. And I've already made sort of a little bit of an attempt at the RNA cannot pandemic thing because of this little thing that we've sort of broken down here. That they have told you that all of the animals with 4 legs and eyes in the front, all of the animals with 4 legs and eyes in the front just use DNA to translate to RNA, to shove through a ribosome and make proteins, and everything that 4-legged animals with eyes in the front are is because of those proteins. You fell in love with the first person you fell in love with because of those proteins. You hate the dog across the street because it wakes you up at night because of those proteins. That's all you are. Proteins in a chorus that results in love and addiction and desire. It just all comes down to physics and chemistry. And more importantly, this physics and chemistry. DNA to RNA to protein. You got no control, no choice, no nothing. And we know from Joshua Lederberg and his student Samuel Epstein that that's bullshit. This book that I shared with them is another Epstein book. It is a book which catalogs 2 successive NIH conferences on gene expression. In relation to what? Not development, not food, not exercise, not daily training of high skill eye-hand coordination, but about permanent changes in gene expression based on first LSD. Then 2 years later, they decided to expand it to LSD plus some opioids and some other drugs that have all kinds of other effects besides addiction. And more importantly, they knew at this time that addiction is gene changes, which is very important for me to express to you that there is no way that anyone you love or anybody that you hate can be blamed for being addicted, because once you are exposed to these substances, genes change and you are effed. Some people are less susceptible to this gene change. Other people have that gene change in a single exposure. Those are all very interesting questions for the Human Genome Project, and that's why they're described in this book and not in Biology 101. They don't want you to understand that since the release of these chemicals, they have known that they permanently alter the trajectory of the arrow that you are. The kid arrow that you are raising is altered by the exposure to all of these substances, and not altered in a small way, altered in a permanent way, because it's a trajectory through time. It's not a circle. You're not a wheel. You're going in one direction. You can't change that direction. The longer you're going, the more likely you're going in that direction. This book and these, that changes this. Because if the DNA can be turned on and off, if the protein that comes out of the DNA can be changed by post-translational modification that could depend on the drug you just took, and those changes can't be reversed, how the hell is that happening if you're not writing it back into your genes? How is that not happening if somewhere in your body, there is a reverse transcriptase that can keep track of that, move it backwards, forwards? These enzymes are crucial for the things they frickin' know happen. How can we have gene expression change? How can we have genes that show 4 different polymorphisms, but there's no writing backwards? Even if you have an experience like an exposure to an extreme drug like fentanyl, how is it possible that there is no writing backwards? It happens all the time in viruses, but not us. We are left with just this. This book can't exist if that's true. I'm sorry, but it can't. We must be able to alter our genes, at least from the perspective of epigenetics, which everybody agrees with. But right now, the only reason why we can't store some of these changes, even from the perspective of our own trajectory, not to pass on to your kids, but your own the only way that happens is if the DNA is changed. It's not just expression. That's not possible. We don't have all of the explanations to do that. We have the enzymes to change them. It's just that David Baltimore tells us that they belong to the viruses. They don't belong to you. No 4-legged animals with eyes in the front have any of those. Only the viruses do. No bacteria. Think about that. No bacteria either. Just the animal viruses. Isn't that so special? Unbelievable. It's so unbelievable that I don't believe it. So that's that assumption. All of this here is an addition made under the pretense that you can inject. So Erin was telling me about her nurse training. I'm just going to pass this around. Unfortunately, this is the workbook teacher edition to the student edition. I could not find the student edition on eBay. But as you pass this around, I'm just going to put the page in the one place that matters the most. These 2 pages. You don't even really need to, what's most important as you pass this around is to see that this is a nursing hypodermic injection, a programmed unit. So what you see here are like what would have been overhead projector things, or even multiple choice questions. And there's no, oh shit, that might be the test. There's no labels in here like dermis, all the fancy names for the skin. You can just see light and dark. And the dark is where you don't put the needle. There's not a lot of complicated things in this, but this is a nursing textbook, a teacher's edition, that's teaching you specifically that hypodermic injection is frickin' hypodermic, not deep, look, don't go to B, go to A. It's really, really easy. And it's not for just some things, it's for the use of the device. And so somewhere in the 50s, maybe around polio, I don't know, we stopped really giving a shit about injecting in the muscle. And suddenly polio, that has something to do with muscles, doesn't it? Like an autoimmune to muscles or some shit. So anyway, pass this around. Please see. It's hard to find these textbooks, but they exist. You could find them. If you find them, you should buy them. And so the working assumptions from the 1920s, which included injecting things in the skin, that's the only one we've dropped. Isn't that bizarre? We actually cared more about where we put this stuff when we first invented the needle than we do now, even though we know so much more about the body. What do we know about the body? Well, I'm going to do my old thing here. What I was going to do for the course, well, I don't know why that's not on anymore. What I was going to do, I had a immunology lecture that I loaded up from 2022 that I already made, but it explained COVID and stuff, and I was going to try and show it and show you what I got wrong. I don't necessarily think that is any better than me drawing pictures. So I'm just going to draw pictures. And I've drawn this picture before, but what I'm now doing is trying to give you a little overview of what you have in your reading and what you can read tonight at the hotel or when you leave after this weekend. I've already put them in there, enough stuff in there so that when you follow up on these big ideas, you're not going to have to look up things on your own. But oh yeah, that book's going around. But you can already follow up. You can already follow up here. So you know this picture that I've drawn before, and I do really think that learning this picture is not a waste of your time. The reason why it's not a waste of your time is because it's a picture that you can learn to draw and give to your doctor. It's a very simple picture. This is the body, this is the lungs, this is the mouth, this is the backside, these are your intestines, that's your whole body. And it's very important that you understand that somewhere there are sphincters, right? We can close this hole if we want to. We can close this hole if we want to. We can close this hole. But stuff goes in, stuff comes out. These are all barriers that are defying the immune system. Okay. The immune system is defining the barrier. And this is the part that I think we don't teach about the immune system that's very good. But the immune system in most places in the body is a single layer of cells. Sounds really ridiculous, but it isn't. It's one layer of cells. But what's the trick? One layer of cells covered by mucus. On the inside, it's always the same. One layer covered by mucus. And we already know what happens in the mucus. The bacteria are happy to grow there. They're happy to grow there because they can communicate to each other, and the phages can move, and the phages can actually move into your body as well. So when you put food, we're just doing basic biology here. When you put food in, it goes down here. How does the sugars and the fat, how do they get into your body? Absorption, right? They go through the skin. Where do the sugars go? They go in the water. So they go in the blood. What's the other half? This is the part that I'm not saying you, I'm saying biology. It drives me bananas. If you've ever seen a frickin' cadaver, there are as many lymph vessels as there are blood vessels. But when you get a picture in a biology book, it's just blood vessels. But actually, there's blood, because that's water, and then there's lymph, because that's fat. You can't absorb fat into your blood. It gets absorbed into the lymph. And the lymph is, of course, the dwelling place of the immune system. That is what the immune system is. It is a lymphatic system that allows the transport of biological material, not water-soluble material, but biological material that includes phages. They just pass freely because they're allowed to. And because they're allowed to, that means they have access to all your cells, all your mitochondria, all your regular cells. They could potentially alter expression in particular places or in every place. And that is a completely uncategorized interaction here. The interaction between these guys and themselves, and also between you and the body. And what does it have to do? It has to cross the barrier. It has to cross the immune barrier. So what happens and as best I can tell, in a respiratory infection, if there is such a thing, and I believe there is that a physiological change happens in the molecular biology of the microbiome in the lungs, which we've actually defined the role of DNA on. We saw how the bacteria changes to become pathogenic, and that change is governed by a bacteriophage, a DNA. They don't say bacteriophage because they don't want you to know that, but basically that's how pneumonia becomes pneumonia. It's a bacteriophage event. So when that bacteriophage changes those bacteria, then you have this problem here that causes the immune system to react. And what they tell you on the TV and on the news is that there are T cells And they're not wrong because there are T cells and B cells. And B cells make antibodies. And I don't necessarily disagree with any of those things. I think that it's hard to imagine that they have accurately characterized all the different kinds of these cells. That's probably not likely, but you've got to be somewhat simple. You know, there might be more cardinals than I say there are. There might be a couple subspecies, but in general, there are cardinals. So in this case, there are T cells. There could be more or less. But the T cells here are defending now against the whatever activation of that pneumonia. But what else happens? That pneumonia is also getting swallowed. And so the trick here is to realize that every night when you go to bed, they don't talk about this very much either, but in your intestines, there are pockets. And I've included some papers for you to read in the thing tonight or whenever you want to about Peyer's patches. Peyer's patches they have their equivalent in other animals, and they have many different names. But what they are invaginations or big deep folds in the epithelium that have specialized cells inside of them which allow, well, it depends on who you talk to, but if you look at the right article, you can actually hear people tell you that your bacteria go into your lymph. They are not in the lymph like circulating, but they are exposed and floating in the lymph inside of the Peyer's patches every time you go to sleep. So the Peyer's patches are definitely a place where the immune system is interacting with the bacteria in the mucus. Now, what's interesting is that the T cells in the lungs tend to be the kind that Tony Fauci likes a lot because these are the kinds that activate your immune system, and the T cells that are in the gut are actually the opposite. The T cells in the gut are by far and away, they're T-tolerant cells, or I can't remember if that's the only way they call them. I think it's T-tolerant T cells. But the gist of it is that these T cells and these T cells are competitive. And they have identified that there are T cells which activate other cells. There are T cells that turn those T cells off. Well, the interesting thing about what I find in the organization of the immune system is the T cells that turn things on are on the top. They're also on the outside. The T cells that turn things off are down here in the Peyer's patches. T cells that turn things off are also the cells that don't work quite right when you're allergic to food. So what's really interesting in my mind is that this isn't even a hard puzzle. I don't have to be an immunologist to understand that if the immune system gets activated here and the immune system can be turned off here and they're connected by mucus, then it makes pretty much sense that if there's an alarm bell here, it can be turned off here. Why would you turn it off here? Well, if the proteins that are being produced here are going down here, if the signals that are being produced by the immune system are also being carried down here, then you're thinking of this as one harmonious thing that moves through time. This inflammation is here before it gets down here. And so there's a response through the system where you have a feedback loop that even works on your food. This is the reason why babies that are exposed to food, I think, after an immunological event like an injection can potentially develop allergies. And it's one of the things that they've scripted the anti-vax people to kind of say. There is 5 different versions of this peanut allergy book. First written by a lady, then written again by that lady, and then written again with a foreword by RFK Jr. So for like 15 years, she apparently understood that if you expose kids to intramuscular injection, then they could become allergic to the food they eat afterward. But she couldn't get to the point, just like no one else can get to the point, that the problem is putting it here. What I will bring tomorrow, which I forgot to bring today, I'm really sorry about that because I was going to have this be kind of a cool thing. I have 2 encyclopedias from the early 1900s where you can look up smallpox and just see a description of it. You can look up a description of the smallpox variolation where they scrape the cowpox into the skin. So that's, again, remember, that's in the skin. Remember that the measles vaccine is actually a subcutaneous vaccine. And so you actually see very quickly, they actually only have one newspaper ad that said that people that were taking the smallpox vaccine were against it. There's only one. It's a very old ad. It's got a picture of a cow person, like a person turning into a cow. And we're supposed to believe that that is enough for you to accept that people did not like this and it didn't work very well. What I find strange is I have 2 encyclopedia sets at home that are 4 volumes from 1920. That's a lot of work to print a book in 1920. You look up smallpox, one of the things that it doesn't say is it's deadly. It's not deadly. It doesn't say that it kills people. What it says is that people became scarred. What it says is people who got variolation became scarred. It says that people wore makeup to hide the scars from frickin' smallpox. It doesn't say that people were dying of smallpox. So what I find strange is that while Paul Offit and Tony Fauci will tell you that smallpox vaccine didn't really work, it was terrible. It was a really bad vaccine, and people were really skeptical of it. Look at this newspaper ad. It shows people turning into cows. If that newspaper ad didn't exist, they wouldn't be able to tell you it. They wouldn't be able to tell you that nobody had any problem with this. If they had a problem with it, they had a problem with it for the same reason that doctors knew everybody had a problem all the time, because it hurts. The most important reason to start injecting people in the frickin' muscle is because your skin reports the damage. Your skin reports the damage with itch, with pain, with swelling, with reaction that is absolutely and totally temporally correlated with the exposure. If you inject a ham sandwich, ground it up and injected it into your skin, chances are really good you'd get a reaction really fast. You take that same ham sandwich and inject it into your muscle, it might take 3 days for you to notice any difference. And by then you won't connect it because Paul Offit says that intramuscular injection is fine. Now I know that this is not really a great, how could I say, a detailed lecture, because I want you to do this on your own, but I want you to have the right idea, because if we were to take the paper that I gave you and you see the diagrams of the cells and how they're arranged in the Payer's patch, you can compare that arrangement to the arrangement of the layers of the cells in the skin. You can compare that to the arrangement of the mucus and the cells in the vaginal walls of every woman on earth. And the arrangement will always be the same. The mucus on the outside, a single layer of epithelium, and then a whole layer of immunology ready to respond, T cells and B cells, just waiting to see what gets through or what the epithelium lets through. And that arrangement is here on the outside, it's here in your lungs, it's here in your throat, it's here in your belly. It's everywhere in a slightly different orientation with a slightly different set of players, but nonetheless, the most important thing to see is that there's always an arrow pointing out, an arrow pointing out, an arrow pointing out. That's the orientation of the immune system. And it's always oriented in such a way so that it can govern what bacterial, biological signals are able to be tolerated and what ones aren't. Where do animal viruses fit in here? How is it that the bacteria just get out of the way? Let them move their way through. And that's what I think should be the most compelling thing here. That, remember, I said this in a break too. They did an experiment in the early 2000s. It is a Spanish group. They created a DNA nucleic acid detector that could detect nucleic acid chains up to 25 nucleotides or something like that. And what it was this big plate of metal, and they had charged electrodes on it and some coating of a chemical that only reacted with DNA or RNA. And they put this thing on the top of a mountain in Spain, and they were able to measure some ridiculous number, an order of millions per day of events where larger than 10 nucleotides of DNA hits this detector. The only attribution that they can give it to is bacteriophages. Could also be, I guess, animal viruses, but the consensus among these people is that what they're looking at are bacterial viruses in the air. Millions of them per day on this small 1 meter piece of metal in the mountains of Spain. So what's happening in your gut? What's happening on your skin? What's happening on your skin when you put sunscreen on versus soap versus alcohol? These are considerations. I mean, I give you an example of how dumb I am. My wife was in the Netherlands last year, and I have this little piece of eczema on my skin, and sometimes it gets infected. And I decided that it would be smart to put bleach on it because I didn't have anything else to clean it. And what that did was it chemically burnt, and then it became cellulitis. So I had to go to the doctor, and the doctor was like, "The bleach is not a great idea." But that was bacterial. It went in. I'm not particularly smart in that sense because I thought, whatever, bleach is bleach. It'll kill stuff. But what I didn't realize, and what that guy explained to me, was you chemically burned your skin. You completely neutralized any protective power that your skin had with that bleach. And it started to make me understand that, and it was kind of this thing that led me to see this, that one of the things that these people have left us with here, with this, and one of the things that is now on the news right now, that bastard Robert F. Kennedy Jr. and that frickin' Jayanta Bhattacharya and every other idiot on the planet is claiming that this was a lab leak, that they actually made the RNA. Now, the most important thing that I can tell you about this particular story that we're talking about, and I'm off topic now, I don't care. And this is really the most important thing. Remember that these books about bacteriology, Joshua Lederberg, Delbecco, Max Delbruck, all these guys, have discovered the basis, the evolutionary basis of heredity in DNA, and specifically all the experiments were done in bacteria with bacteriophages that are carrying the DNA. And so, what did I just say about coronavirus? Just help me. Why did I go there? The what? Oh, sorry, yes. So in that, yeah. Sorry, my bad. I'm sorry. I had a space cadet there for a minute. So in that same time, the way that they figured it out was that they just stumbled upon the fact that bacteria, they don't have chromosomes like everything else. One of the things they figured out is bacteria actually just have little circles of DNA. That really confused them because they didn't know why it would be in a circle. And so for a while they were kind of stuck on that, but they understood that those circles, when the bacteria divided, it would copy those circles before it divided. So that was another sign that it was using those circles to carry information. So what happens crucially at this moment in time is that recombinant biology is made, because these people in bacteria realize that if we take this DNA from one bacteria, we can have this other bacteria make lots of copies of it. And this is really the heart of the Human Genome Project. It's the heart of all genetics. It's the heart of everything that has to do with this stuff. Because if you can make a lot of DNA, then you can sequence it. If you can make a lot of DNA, then you can put it in places, you can find it, and you can test it. The only way that they could do this is with bacteria. So they can make a lot of DNA or a lot of RNA, and they can put it in places, and it does stuff because of that. Because RNA translated into protein does things. And so they have discovered this. They have produced this. But what they have told us is that this thing all by itself, if we make that, it can do all kinds of things by itself. Now, what is so hard to express here is that that's so close to the truth. It's so absolutely close to the truth. But it's not quite right. Because what they actually are doing, I have to take my pretty picture down here. How many people think they know what PCR is? The most important invention ever? Anybody? Everybody knows what PCR is? PCR is the polymerase chain reaction. It allows them to, another reason why you should know Joshua Lederberg is, I'll take this one. I know this is going to not really blow anybody's mind, but that's Joshua Lederberg. And more importantly, that's Carrie Mullis. And I would bet my life on it. So what you have here is the history of PCR in the Muppet Show. The most intelligent scientist ever to live in America has been characterized as Honeydew right here. And then this guy, bee, 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 bee. Well, what actually does this describe is exactly the description of PCR. What happened? This guy, some stoner LSD users driving down the street one late night, and he says, oh my gosh, I got a great idea. He gets home and he writes it down. What he says is that double-stranded DNA can be heated and it separates, and then if we put DNA polymerase in there with a little piece, we could copy this one and copy this one, and then we'd have two pieces. And then if we took those two pieces and we heated them up, and then put little pieces in there and let the enzyme fill it up, we'd have four. And he supposedly had this idea while he was driving home in a car, and then he wrote it down, he came to work. And he tried it, he put some DNA in there, and then he heated it up, and then he added some enzymes, and then he cooled it down, and then he filtered out the enzyme, and then he heated it up, and they put the enzyme in again. And it was a real pain in the ass because you had to take the enzyme out before you could heat it, because if you heated it with the enzyme in there, the enzyme would get ruined and then it wouldn't copy. So every time he wanted to make a copy, he had to pull out the enzyme and put that back in. Now, we're to believe that he came up with this idea on his own, and then he put it on a poster, and the first person who saw that poster was Joshua Lederberg. Joshua Lederberg knew exactly what he had found. He had found a way to make lots and lots of DNA, which is something that they only knew how to do in a bacteria, and more importantly, only with the shit that was already in the bacteria. You could make a bacteria make more copies of shit that was already in the bacteria. Now they had a way to put other stuff in the bacteria, have the bacteria divide, and lo and behold, they copied it too. And we haven't come any farther since. That's all we got. That's how the COVID vaccine was made. That's how virology is done. I want you to think about the fact that you can go on PubMed right now and you can search for the term infectious clone. It's a dumb name that comes from clone, which is the many copies of, right? And infectious because David Baltimore and Vincent Racaniello, two very fake molecular biologists, took the polio virus as a sequence and used bacteria to make lots of the DNA. And when they put that DNA into a cell culture, they claimed it made virus. Probably did. I don't know. But the point is that experiment doesn't exist unless they can take that DNA and make a lot of it in bacteria. Without that, it doesn't happen. What I want you to understand is that if you go on PubMed, the big website for all the papers, and you search for infectious clones, you will find hundreds of thousands of papers where people claim to be doing virology, but what actually they are doing is they are making a large quantity of pure DNA using a bacteria. Sometimes they're actually just ordering it from a company that does that. And then they're taking that DNA and they're putting it on a cell culture, and what happens is called virology. It could be that it gets packaged, it could be that the cells get destroyed, it could be that they grow, but whatever it is, that's virology. But virology is actually taking DNA that is made in a bacteria and apply it to another model. Put it into the throat of a monkey, put it into the lungs of a mouse, inject it, put it on a cell culture. If you start with DNA that you made in a bacteria, you're doing virology. That's what they do. But it's not virology, it's molecular biology. It is studying this, the central dogma, the RNA to DNA to protein. It's just that now they can push from here, or they can push from here and see what happens. And I'm not saying it doesn't work, because it does. I used it in my lab all the time. All academic biologists are ordering RNA and putting it in their animals to overexpress a protein. I said earlier, expressing a protein is when a gene or its RNA equivalent are translated through a ribosome. It's called expression. And so when you express a gene, you are translating it to RNA and eventually turning it into the protein that it encodes, and that's expression. And so if they transfect you, they are expressing a protein in your body. And that's what they claim they did with the COVID vaccine. They expressed a spike protein in your body, and then your body reacted to that expression. Now, the important thing to understand here is that I guess I didn't write that. That wouldn't be possible without intramuscular injection. You can't transfect somebody by getting them to drink it, because if they drink it, then not only does it go through past the microbiome and the mucus, but it's also got to go through the liver, because everything that goes through your gut first gets filtered through the liver. And so there's all kinds of things that aren't handy for that reason. You can't study gene expression by giving people food. So they had to get rid of it at some point. They had to get rid of this to make you into the experimental animal that they needed. They have to get to the point where they can push from here, And right now you can hear the push. The push is extreme scenarios. In the 80s, the extreme scenario was Jesse Gelsinger not expressing a certain protein in his liver so that he couldn't digest starch. And so they used an adenovirus to transfect his liver to get him to express an enzyme that he didn't have in hopes to cure that. The problem is that they made him express a protein that his body didn't have. So it's a foreign protein to his body, and sure enough, his body destroyed his liver and he died. That was the first transfection experiment of the Human Genome Project, and many of the same people that are involved in the, measles, anti-vax, whatever bullshit, we're involved in that too. And that doesn't make any sense. It doesn't make any sense unless it's all part of the same, forcing these bad biological working assumptions on everybody so that everything is either stupid complicated or dumb simple. And so you're not left with any questions because you don't have anything to hold on to. And so I'm suggesting that actually the high school teacher is the place where we can win, because the high school teacher is the one that can still inspire the awe that will prevent this from becoming a working assumption. It will maintain the awe in the irreducible complexity that will keep this working assumption going. So if the title of this last little talk is immunology, virology, and hypodermic injection, then what I'm trying to sort of impress upon you is that Joshua's microbiome, it's his word. Joshua is the guy they quote. Joshua Lederberg, this guy, is the guy that they quote in the script of Outbreak. And he's the guy who wrote the virology review in the script of Outbreak so that people that were reporting on this movie could tell you exactly what it was about and why it was so important. He wrote that. And the quote is him in the beginning of the movie. And the quote is, "The single biggest threat to man's continued dominance on the planet is the virus." That doesn't sound like something Josh would say without a tongue-in-cheek or whatever the right expression would be. He knows that's bullshit. He knows that's a national security myth that he's telling you in a frickin' Hollywood movie with who? With who? Dustin Hoffman? Frickin' Morgan Freeman? Kevin Spacey, Dustin Hoffman, Cuba Gooding Jr. How many more stars we need to have in here? Rene Rousseau, it's everybody that was popular at that time, all in one movie about a virus outbreak that starts with Joshua Lederberg at the start. And so I guess what I'm trying to summarize a little too early is that I don't think you need to defend yourself to the doctor. I think what you need to do, and you don't need to defend yourself to your high school teacher or to your college teacher. You just need to understand the complexity in such a way so that you can express it simply. You need to be able to say things like, what RNA can't pandemic? That doesn't make sense. Intramuscular injection isn't very smart if you know anything about the immune system. If you say that to a doctor, most of the time they have a very bad reaction to it. I actually, in 2020, wrote a 5-page review trying to explain that to my doctor. And he told me that he didn't have time to read all those papers. I'm sure they're pretty good, but he doesn't agree with me. That's how quick they can dismiss it. And so you, as a young person, this is especially for you guys, it's impossible for me to understate how the COVID pandemic was for you, because they let you go back to college. They let you make all those decisions on your own, away from your parents or your peers, let's say it that way, so that essentially the first round, you're not aware of this, I'm just going to throw random things out here until the end. You're not aware of this, but at the University of Ohio, in 2020, there were faculty members who became aware that the University of Ohio had a contract to sell the remnants of COVID vaccine tests or COVID positive tests, whatever, COVID viral tests. They had a contract to sell the remnants of those before the pandemic. It was arranged. And faculty members found out about it and started a lawsuit, and there is still a lawsuit going on right now at the University of Ohio for this, but I think it's going to get burnt because they have too much resistance from the rest of the university. I can assure you, though, that what's happening right now, as we said with chess and Go, is that if you realize that they need this data, from you, the DNA from you, they needed it from you. They actually already did the first iteration, because when those college kids went back to university, they were in the college health system, and systems like Ohio, for example, are in the EPIC database. Now, what I want you to see is that the first iteration is really very simple. They have a perfect check. The game can be won. How's the game won? Well, they take those two guys, EPIC database, and then they look at all of the genetic samples that they have from the COVID time, and they see if anything pops up that lights up with them. And if it does, that's actually a winning game. And if the more times they are able to match up what looks like a half, what is it, Thailand? Thailand? Is that what you said? Taiwan. Yeah, half Taiwanese, half American, and there's only 8 of them, and they all went to that school. Holy cow. Now you see where they're already at. They're building data centers in America, the place where the most remnants were created, the place where most remnants are still being created, in order to do iterations of this test. They have to see how easy is it, because they already have de-identified data. They weren't allowed to keep your name on there. All those things are randomized. So they've already got the perfect setup. They've got all the data from all the students, and then they have a randomized set of frickin' tests. And so if they do it right, they're going to get the best matches, and they can do it again and again and again. And 3 iterations later, 80% of the students can be matched up without even needing to know their names. They're already working on this, I can assure you, because this is just the first iteration. What they need to do is get it right before Freddie has kids. Again, I need you to think about Mike and how Mike doesn't have very much time before he's old and dead. And so Mike's got to organize the puppies and make sure they know what they're doing, and only the puppies that matter. And they've been doing this for us already, the parents of these people. That's why Mark's discovery is so important. If I would have known that Mary Holland's brother was working for fucking Tony Fauci when I was working for them, I would have gone to the New York Times. But instead, I wasn't. Mark's frickin' son was murdered, and I was paid $108,000 and given access to like 4 different doctor's offices' records and told you had to work on this shit and show up in this Zoom meeting. And you're a hero because you figured out it's a lab leak. We're going to write a book. And Mark's not calling me. I don't know why. Can't really get any word out of him because he's not exactly proud of what's just happened in his family. So I just keep working for Brian Hooker, who says at some point in our little thing that I think Mary's mom worked, or Mary's brother worked for the CDC or something like that. I don't know. So he even lied to me about that. And it turns out that Brian Hooker, the scientific advisor of CHD, was actually a person who used recombinant DNA to transform plants to make fucking medicine, excuse my language, already for 15 years before he had an autistic kid. Then he had an autistic kid and became part of CHD. Now, I'm not saying he doesn't have an autistic kid. What I'm telling you is he's a molecular biologist from the Department of Energy who now claims to be a biologist at Simpson University. And he's not. He's a guy who used to work for the Department of Energy, the same Department of Energy that governs the frickin' Human Genome Project. The same Department of Energy. He was transforming plants to make medicine for 15 years before he became the anti-vax leader of CHD with anthrax Meryl Nass and frickin' NIAID Mary Holland. And her mom, who worked at the same cancer hospital that Tony Fauci's dad was the pharmacist for 40 years. The pharmacist that was producing the drugs that they treated cancer people with, that included fentanyl. These are all just, it's not possible anymore. It's just unfortunate that I got stuck with this now instead of 3 years ago when I could have done something more. Right now, I know for sure that the problem is that I was in those organizations and tried to teach them this stuff. I've had this kind of a conversation with this level of energy with Robert F. Kennedy Jr. on Zoom with other CHD employees listening. And I don't know what to say other than they ignored me. It's not like they said you're wrong or I don't agree with you or whatever. They just said we're not doing this. We're building a big top tent of health freedom and we don't answer questions. This is bad because it is going all the way down to a high school biology level of distortion, and it's not the high school biology teacher's fault because you can't expect them to be able to see through that level of consensus. And that's what it is. It's a consensus. It is a consensus about things that are not true based on the fact that a long time ago it seemed plausible. I'm almost done. If you were in the 1920s and somebody said that in the future we're going to have spaceships and they're going to go warp speed and we're going to be able to teleport people from spaceship to spaceship, it wouldn't seem that ridiculous. In fact, when you watch Star Trek now, it still doesn't seem like 2 millennium from now. Why not? But I think now at a certain moment we've come to physics where we understand that's probably not going to happen. We're not actually ever going to transport anybody to another place instantaneously with some sparkly lines that go through it. And Scotty's not going to say, oh, I can't keep you in here, I'm losing your pattern. That's not, that physics was imaginary. And if a physicist stood in front of you right now and said, I hate to break it to you, but that was probably not a right prediction. We're not going to do that. But on the other hand, if a biologist says to you that, well, we predicted that we were going to cure all these diseases because we were going to take over the control of the genome, we got that slightly wrong. You don't hear anybody say that at all. But I have an article on your thing of Joshua Lederberg saying it in 1993. That after all these years, Joshua Lederberg writes in a newspaper that, well, it's kind of surprising, right? After all these years, all these promises, not really much happened. And he gives one example of what happened, and that's it. Everything else is still just talk. So you can find that one in there. It's 1993. It's a great article to read. I'm probably going to start there tomorrow with my stuff because it's like a nice history of the discoveries of genetics and why they're important in the words of Joshua Lederberg. And at the end of that, he talks about the limitations without saying what he should say, without saying this stuff that I've said, again, because I think, and I think Mark would agree with me, that at some moment Joshua Lederberg, he was part of the group. You get into the room with people and they make decisions and you give them ideas and then they go with it. And at some point in time, they figured out how to manage the population pyramid. They figured out a story that they could tell people that would make sense, and they went with it. I think it's even possible that it's a little darker. I think it's possible that they had a very moral and very what's the right nice word for nice? They tried to come up with the most humane way of managing our population pyramid. That's why culling. That's why here there's a Green Lantern, Green Arrow comic from the 20s in New York City that says the needle's more deadly than the atomic bomb. They knew. They already knew that one of the humane ways to manage the population would be to, give them a painless way to go out. Now, if I end here, I want you to see that Mark and I had only just figured this out very recently, but actually the inverse is always the truth, and we've never really said it so succinctly as that except the last couple months. And the inverse in biology is also pretty much where we're at right now. They tell us that gain of function research can create a special RNA that can do things that all other RNA cannot do. But what they don't tell you is that gain of function chemistry can make an opioid that's 100 times stronger than morphine, and then again make another compound that's 100 times stronger than that compound. Gain of function chemistry with fentanyl and carfentanil. And those numbers aren't fake, those numbers are real. They're real in terms of how they bind to proteins, and they're real in terms of how much you need to kill. I don't know if you watched that video that he recently posted, but I just learned that fentanyls were created as giant animal tranquilizers. The original use was that you only need a few nanograms to knock down an elephant, and that's what they were useful for. They're so powerful that you can't take carfentanil and, dilute it enough for it to not really kill you. It's that powerful. There's no reason for that drug to exist. It doesn't exist in nature. It is something that we designed by chemically choosing things that fit the holes better, etc., etc. That's real. America, as Antonio just showed you, is under attack by gain-of-function chemicals, and at the same time, our NIH director, a guy who I had tacos with 3 times for reasons I can't explain, says that gain-of-function RNA is what we need to be afraid of. And I think that's as inverse as it can get. It's as inverse as it can get. And so I don't know if I'm ending on the right note. I'm not leaving now because, it's 7:30, but you could, if you're tired of this, can leave now and show up tomorrow. And if you have any questions or anything you want to talk about, we can talk together or a group. I just have the feeling now I'm going to keep talking for no reason, so I'm just going to stop now and see you in the morning. But I'm not going anywhere. Thank you. Thank you.